Hi, this is Kim, and today we are starting to talk about the animals. Remember that animals are in the domain Eukarya, so they are eukaryotes. Remember that this domain includes plants, animals, fungi, and protists, which were the single-celled eukaryotes. So they have complex cell structure, membranous organelles, DNA is enclosed in a nucleus, all those eukaryotic cell characteristics. Remember that under domain, the next level of classification is kingdom. And there are two kingdom names for animals. Kingdom Animalia, which is the one we're going to use in this class. They can also be called kingdom Metazoa. But we're going to stick with Animalia for this class. Under kingdom is the classification level phylum. And we are going to go through the various animal phyla that have the most number of species. We're not going to cover all the current animal phyla, but we are going to include those that have the most species. You do need to know each phylum name. So as we go through each phylum, we are going to look at examples of organisms in each phylum. We're going to look at characteristics that they all share and any advancements over the previous group. And you need to know all of that. Under phylum, in order of classification, is class. And I will be giving you some class names in some cases, just to show you the diversity of that phylum. But you do not need to know those class names. We're just giving those as examples. It then goes order, family, genus, species. We will not be covering any of that for the animals in this video. Let's talk about the characteristics of all animals. And I will be showing you some slides too. I just really wanted to start with you taking some notes, and I think the best way to do that is for me to write as if I was writing on the board in the classroom. So characteristics of animals, they are multicellular, meaning they don't consist of just one cell. They are all heterotrophs, which means they need to eat their food, and in the case of animals, they ingest their food, and most of them digest that food in a cavity of some sort. We're going to talk about body cavities and the significance of developing a true body cavity in animals. But all animals do some type of digestion in a space inside a cavity. Their cells do not have cell walls. So what that means is on the outside of the cell, you just have the plasma membrane. No cell wall around that plasma membrane. Recall that plants, fungi, and bacteria all have a cell wall around that plasma membrane, but animals do not, and that's one of the characteristics that defines animals. Animals have tissues, all but the sponges. And tissues are a group of cells working together to perform similar functions. And you had this definition at the very beginning of the semester when we talked about the levels at which we study biology. So for example, you have skeletal muscle tissue, and it's a bunch of cells of the same type that work together to perform the function of muscle contraction to move your body. And that tissue type is actually called skeletal muscle tissue. Within that skeletal muscle, you also have blood vessels and nerves and other types of tissue that are performing different functions to make those muscles move. So that's what tissues are, and that is a characteristic of animals. And all but the first group of animals that we study are going to have that. Most animals have some type of symmetry. And there are two types of symmetry that we're going to talk about when we look at the animals. There's what's called radial symmetry, and there's what's called bilateral symmetry. Radial symmetry means that you usually have a round organism that can then be divided in a number of ways so that the two halves are identical. So if we divided this organism this direction, these two halves would be identical. We could also divide it this direction or this direction. And in all of those directions, we could get two symmetrical halves. Bilateral symmetry, on the other hand, means you only really have two planes that would be identical if you cut the organism. So there's a specific way that you would have to look at it. So for example, in this organism, we could cut it in half, and the two lateral halves, bilateral, would be mirror images of each other. So that would be bilateral symmetry. All but the sponges are going to have symmetry. So that's an animal characteristic, again, except for those most basic animals. In fact, sponges are sometimes not included as true animals, and we'll talk about that in a second as well. When we get to the body cavity, there are going to be some true advantages to having a body cavity. So I'll be coming back to the notes to really write those down for you when we get to that part of the lecture. So let's start though with the sponges. And I'm going to write some characteristics of sponges, and then I'm going to go to the slides and show you examples of sponges. So this is going to be our first phylum name. So again, we're in domain Eukarya. Kingdom Animalia, or Metazoa. And then phylum Porifera, and this includes the sponges. Sponges are the most numerous animals in the deep ocean, if you can believe that. There are at least 2,000 species of sponges, could be higher. And they're sometimes not even classified as animals. Some classifications call them parazoa, meaning they're right on the boundary of being true animals. And the reason for that is they don't have any true tissues, and they don't have any symmetry. Meaning there's not a plane at which you could cut one of those sponges and have the two halves look the same or be mirror images of each other. They have larvae that are the juvenile form of the sponge that are free swimming. So they do have locomotion, but the adults, for the most part, do not move. They're what's called sessile or sessile. That means not moving. And you'll see this term with some other organisms that we talk about. So that's an important term to know. Because they don't have tissues, they really don't have any organs. But they do have some complex cells working together, not as true tissues, but they work together to perform some really important functions for the sponge. So let's look at a picture of a typical sponge. Sorry, I'm going to go to her. slides here. So this is a sponge, and they're right here at the very beginning of our PowerPoint presentation. So not moving as adults. They're very beautiful. Some can be really large in size. Here's another example. And another one. Some can be even bigger than a person. And let's look inside the sponge for a minute. There are these cells, choanocytes, and that means collar cells. And they have a flagellum, that little tail-like structure, and the flagellum moves and it brings in a water current through the pores of the sponge. So water's going to come in through the pores and go out through the top, called the osculum, which also means mouth. And that water current is generated by the flagella on these collar cells moving. There is mucus associated with these collar cells, and they're able to trap debris and small organisms so the sponge can eat. So a group of cells not really forming true tissues, and actually they look a lot like protists. So it's almost like a bunch of protists kind of got together and formed this organism, and they're working in a cooperative fashion so this organism can carry out its basic functions. Associated with each collar cell is another type of cell called an amoeboid cell, an amoebocyte. And you can see it actually looks like a protist also. This is where digestion is going to occur for the sponge. So these cells are right up here working cooperatively with the collar cell to trap food and then digest the food for the sponge. It then gets dissolved, dissolved. It gets absorbed into the other cells of the sponge. Also, sponges have these spicules, which if you've ever touched a true dried sea sponge, not like a sponge you buy at Target that's blue for washing your dishes, but a true sea sponge, they're kind of pokey when you touch them, and that's the spicules. That gives support, so they're sort of like skeletal elements for the sponge. So sponges are more complex than you would think. But again, they don't have any true tissues, and they have these groups of cells that really work together in a cooperative fashion to carry out the functions of the sponge. The next group that we're going to look at are the cnidarians. But first, let's go back to sponges for a minute. So we're still in phylum Porifera. And let's write down some other things they have. They have collar cells that create the water current. And trap food. And then those amoebocytes digest the food. Spicules are the skeletal elements that give support. So those are the sponges. The next phylum is the phylum Nidaria. So this word starts with a C, C-N-I-D-A-R-I-A, Nidaria. The C is not pronounced. And this includes a lot of different organisms. This includes the jellies that we often call jellyfish. It includes the Portuguese manivore. It includes anemones. It includes corals and hydra. Hydra are freshwater Everybody else is marine, meaning they're in the ocean. So what do cnidarians have in common with each other, and what are their advancements? Let's start with some of their advancements. Cnidarians are the first with symmetry, and they have radial symmetry. That radial symmetry is actually only true at one stage of their life, called the Medusa stage. And we're going to look at that in the pictures. They really have two stages that we're going to look at. So that's going to be under the category of characteristics they share. Not only are they the first with symmetry, they're the first with true tissues. And all of their structures are formed from two tissue layers in embryonic development, the ectoderm and the endoderm. They're missing a third tissue layer called the mesoderm. All animals after cnidarians are going to have three tissue layers in embryonic development that form all of the structures of that animal. So if you only have two tissue layers, there are certain structures you can't form, such as muscle and bone and some other complex structures. So they only have two tissue layers to work with for building their structure. When you just have two tissue layers, you're what's called diploblastic. So all cnidarians are diploblastic, meaning all their structures come just from two tissue layers. They don't have a concentrated nervous system. So they have a very primitive response system. And because they only have two tissue layers, they have no true organs. Again, they can't build that complexity with just those two tissue layers. They only have one opening, and that opening goes into what's called the gastrovascular cavity. Let's look at this word, gastrovascular. That implies digestion and circulation. So several things happen in that gastrovascular cavity. That's where all food is going to get digested. So digestion takes place there. Gas exchange. Waste products are going to go into the gastrovascular cavity. Gametes are going to get discharged into the gastrovascular cavity for reproduction. So those are some characteristics that were advancements over the sponges. What are some other traits that cnidarians share with each other? They all have stinging cells. And those stinging cells have a structure called the nematocyst. And I'm going to show you a picture of that, but if you've ever been stung by a jellyfish, you have experienced the nematocyst. It's coiled up inside these stinging cells and it discharges, and that is what stings you. It's used for capturing prey and also for defense. So when it stings you, it's a defense mechanism. And then, as I mentioned a couple of minutes ago, there are two life stages in the cnidarians. There's what's called the polyp and what's called the medusa. The polyp is sessile, meaning it doesn't move, whereas the medusa is free swimming. Some cnidarians are polyps as juveniles, and then the medusa stage is the adult. In some, it's the other way around. Okay, so for example, in corals, the juvenile stage is the medusa, but the adult is the polyp. So the mature corals that you see, that is the polyp stage, obviously not moving. The jellies, it's the opposite. The juvenile is actually not moving, but we've all seen the adults, and they're the medusa. Okay, so now let's quickly look at some pictures of the cnidarians. Sorry. Goodness me. First, I want to show you this slide. This is just showing you those 3 tissue layers that we're going to be really looking at in more detail in a couple of minutes, but it includes the ectoderm, the mesoderm, and the endoderm. And you can just see some examples of structures that can develop. So if you only have 2 of those layers, you're just not going to be able to develop the complexity. So the fact that cnidarians only have 2 of those tissue layers means they can't really develop a lot of those complex structures that you see. So cnidarians just have ectoderm and endoderm. They don't have mesoderm. Okay, this is who they include. The hydra are the freshwater. Everybody else is marine. So beautiful jellies, anemones, coral. So these are living organisms. These are the polyps. This shows you a good picture of that nematocyst. It fires and stings you. And this shows you the 2 different life stages, the polyp and the medusa. So the polyp doesn't move, the medusa is free swimming. I'm gonna go ahead and show you this picture of a flatworm. See how flat that worm is? Really big surface area relative to volume. And that's the next group we're going to look at, the flatworms, Platyhelminthes. Let's go back to the notes. Phylum Platyhelminthes. So this is our 3rd animal phylum. Platy means flat, and this actually means helmet. So these are the flatworms. What do I mean by flat? Well, if we cut one of these worms in cross section, they're very flat relative to the roundworm. So this would be a roundworm. The reason for that is they don't have a body cavity. And because of that, they can't develop a complex circulatory system, and they can't move materials such as nutrients, gas, waste. It all has to take place by diffusion. So move materials, oops. By diffusion. So let's think about that. If you have oxygen out here and it needs to diffuse to the middle of this organism, it's going to take a long time. Versus if you're flat, that's pretty simple, relatively speaking. So large surface area relative to volume. So they can absorb gases through their skin, and those gases can diffuse through all of the cells. Platyhelminthes are the first with 3 tissue layers. So they're called triploblastic. That means they have endoderm, ectoderm, and mesoderm, which means they can develop more complex organs. But they can only get so complex because they don't have a body cavity. Not having a body cavity is called being acoelomate. A coelom is a body cavity. So A means without, and coelom is the body cavity. When we talk about the advantages to having a body cavity, it will make more sense why it's a big deal to not have a body cavity. So that's coming up soon. So first with 3 tissue layers. So they can develop more complex organs, but again, they can only get so complex because they don't have a body cavity. They're also the first with bilateral symmetry. Which means I'm going to draw a planaria worm. And we can cut this worm in half, and the 2 halves are mirror images of each other. So bilateral symmetry. They can only, again, get so big volume-wise, otherwise diffusion couldn't happen. Most of them are parasitic, meaning they have to have a host in order to survive, and they have some complex adaptations for parasitism, such as hooks and suckers for holding on to the intestines if that's where they're living. They've developed strategies to not be detected by your immune system. A lot of them don't have eyes or any way to see. No point in seeing inside the intestine of a host. Very thin outer layer for just absorbing from the host. A couple of terms associated with being a parasite that are important to know. An intermediate host is a host in which the juveniles, usually the larvae, live in this host. And then the larvae are transmitted to what's called the determinate host. And that's where the adults develop and reproduce. And eventually the eggs are going to come out of that determinate host. If that parasite is transmitted by another organism, that organism is called a vector. This is an organism that transmits a pathogen. So a lot of vocabulary here. What is a pathogen? A pathogen is a disease-causing organism. And in humans, that includes a lot, including viruses, which aren't really living organisms. But in humans, the list of pathogens includes viruses, bacteria, protists, fungi, and worms, including the flatworms. So what type of flatworms are parasites of humans? They're mostly tapeworms, which I'm sure you've seen pictures of before, but I'm going to show you some pictures of tapeworms, and flukes. Here's one more characteristic of all flatworms, and that is that they have what's called an incomplete or closed digestive system. So even though they have an intestinal tract, it's somewhat inefficient because they just have a mouth, no anus. What does that mean for this worm? So here's the worm. And food's going to come in through the mouth, and it's going to go through the gut, and then it has to come back out again. So food goes in and waste comes out. And that's pretty inefficient. Think of it this way. If you have another opening to your digestive system and food can go in the mouth and then eventually reach the intestines, we can increase that surface area for absorption by coiling, folding, and eventually the waste is going to leave the body, but that's going to give us a much more efficient way to digest and absorb the nutrients. In the flatworms, they don't have that option. Food comes in, the waste comes out the mouth. Okay, let's quickly look at some pictures. These are the beautiful marine flatworms. Again, you can see large surface area relative to volume. Same here. Here's a tapeworm. This is a picture showing you the life cycle of the tapeworms that infect your dog. So you can see that the eggs come out in the feces, and then the eggs are actually eaten by the juvenile fleas. The way your dog is going to get that tapeworm is it's going to eat the flea, and then the tapeworm is going to mature inside the dog. So the flea is actually the intermediate host, and the dog is the determinate or definitive host. There are two terms that are associated with that. It could be called the definitive host or the determinate host. Sorry, I was going to draw that on there, but it's not letting me. This shows you some of the adaptations in the tapeworms that we talked about before. You see those hooks on the end, the suckers for attaching to the intestines, no eyes. These are planaria. They are free-living, non-parasitic flatworms. This shows you some of the complexity. The arrows here are kind of showing you the two ways that the muscles can contract. So longitudinal muscles are going to be able to contract him front to back, whereas circular muscles will contract around the body. You can see there's a nerve cord, ovaries, testes, intestines. So they are able to develop some more complex structures because they do have 3 tissue layers. These are flukes. This is a schistosome worm, and almost 1 million people a year die from these worms in third world countries. They are very dangerous. So that's a picture of them. Okay, I'll go ahead and show you the next group of worms while we're here. The next group of worms are going to be the nematodes. These are the roundworms. So I will go back to the notes and write this down for you. This is a microscopic nematode found in the soil, non-parasitic. They say you can take a cup of soil and have over 1 million nematodes in some parts of the country with really fertile soil. So this would be a non-parasitic roundworm. But then there are parasitic roundworms, hookworms, which are found in the United States. Trichina, which is found in muscle of the pig. So we eat the pork and we get these parasites when you don't cook your pork all the way. You can also get ascaris from pork, which is a parasitic roundworm. Filarial worms are parasitic. They block the lymphatic system and cause elephantiasis. And you can see in this case, the vector is a mosquito. The mosquito is going to transmit that worm to humans. And the mosquito is also where part of the life cycle happens for those worms. So it would also be called the intermediate host. So let's write down some advancements in roundworms, the nematodes. Oops, sorry. Okay, so we've had phylum Porifera, phylum Nidaria, phylum Platyhelminthes, and now our fourth phylum. Phylum Nematoda. And sorry, I don't want to use red. I'll rewrite that in black. Phylum Nematoda. These are the roundworms. They're able to be round because they do now have the start of a body cavity. It's called a pseudocoelom. And they are the first to have what's called a pseudocoelom. So pseudo means false. And of course, the coelom is the body cavity. They can fill that pseudocoelom with water or other fluids. And it forms what's called a hydrostatic skeleton. So now they can get bigger. They have more support. It gives them this kind of jerky type movement. Imagine, you know, those long cylindrical balloons that clowns use to make balloon animals? Imagine one of those not filled with water, just flat, not filled with air or water. And then imagine that you filled one of those completely with water, how much support that would give to that balloon. The same thing happens in the nematodes. They are the first with a complete digestive system. Meaning mouth and anus. This is sometimes called an open digestive system versus a closed. Huge advantage, as I mentioned before, much more efficient in terms of delivering nutrients to the tissues. They do have 3 tissue layers. They are starting to develop more complex organs, but we're not really going to get the most complex organs until we get to the organisms with a true body cavity. So again, this includes the soil nematodes and some other free living examples that are roundworms. It also includes a lot of parasites. And we saw examples of those. The ones that cause elephantiasis, the ones found in pork, including ascaris. So lots of parasites that are roundworms. Okay, that is it for video number 1. In the next video, we're going to start talking about the advantages to having a body cavity, a coelom, and who has a coelom.